I'd like to welcome everyone today to the Vasculitis Foundation webinar. I'm Kathy Olewski, the host for the Vasculitis Foundation's educational webinar series, but I'm also a patient living with MPA vasculitis. These webinars are part of the Vasculitis Foundation's commitment to patient education, and we would like to thank our sponsors, AstraZeneca, Amgen, and Novartis, for supporting the webinars. And today's webinar is about genetics in vasculitis with Dr. Sharon Chung. After we finish the webinar today, there will be a recorded version on the Vasculitis Foundation's YouTube channel, and everyone who is registered will receive an email with a link to it as soon as it's available. But before we get started, I'd like to go over just a few details. Because this is a live webinar, we've muted everyone except our guest speaker. So in today's webinar, Dr. Chung will do a presentation about genetics and vasculitis, and after her presentation, you will all have the opportunity to ask her questions. These questions will be asked by typing your questions into the Q&A box. If you don't see it, click the three dots on the bottom far right of your screen where it says More. And please make sure your question is a general question about today's topic, genetics, as Dr. Chung cannot comment on your specific medical issues. There's also a chat box for you to chat with others who are in the webinar and the Vasculitis Foundation staff, but please remember, use the Q&A box for your questions. And now that all the housekeeping is done, I'd like to start by introducing our speaker for today. Dr. Sharon Chung is a rheumatologist and is the director of the University of California, San Francisco Vasculitis Clinic. She specializes in autoimmune diseases, particularly systemic vasculitis, and Dr. Chung earned her medical degree from Johns Hopkins School of Medicine, completed a residency in internal medicine at the Johns Hopkins Hospital. She completed a fellowship in rheumatology and earned her Master's of Advanced Study degree in clinical research at UCSF. So welcome, Dr. Chung. We're so happy to have you here with us today.
Thank you, Kathy. Happy to join everyone.
And
let's go ahead and see if I can get the IT aspects of things to work.
And while you're doing that, if you have any disclosures, if you would share them with us at this point.
Of course. Hold on a minute. Da-da. Here. All right. A thumbs up, Kathy. Can we see my screen? And does it look correct?
It does.
So, hi, everyone. My name is Sharon Chung. As Kathy said, I am a rheumatologist at the University of California, San Francisco, where I direct the Vasculitis Clinic. I have a particular interest in treating patients with systemic vasculitis, and I don't have any disclosures. All right, I will go ahead and get started, and hopefully this will work. Okay, so I think, the topics for our discussion today are fairly short. I mean, this is a very focused presentation on vasculitis and genetics. And when I was thinking about, what I should talk about, I thought I would focus on the two questions that I get asked the most about genetics. First, is vasculitis genetic? And are there any genetic tests we should be sending off because I have vasculitis? So I'm going to spend almost all the time focused on these two questions, but I do have a disclaimer. I'm not going to discuss every single genetic association for every type of vasculitis that's ever been identified. I think we would be all bored to tears within seconds. I mean, that's not the goal today. So what I've really done is I've picked out examples which can guide us how we think about genetics for different types of vasculitis. And the overarching principles illustrated by these examples can be applied to your specific form of vasculitis. So just to get us started, the question is vasculitis genetic? And I'll be honest, this question means different things to different people. I'm a scientist by training. And so when you ask me this question, is vasculitis genetic? What this means to me is do changes in genes impact one's risk of developing vasculitis? Now, most of the time, when people think about whether or not a disease is genetic, they're thinking about single gene diseases. These are diseases where if you have one change or one mutation in one gene, that's enough to cause disease. And individuals who have this genetic mutation and this disease can pass this gene mutation onto their children, and then their children have an increased risk of disease. Family trees for these types of genetic diseases are often what's seen here on the left, where you have an affected individual. That individual has passed that genetic mutation onto his children, who have passed it on to their children, who then have passed it on further. And the most common examples, or the best known examples of these single gene diseases, include sickle cell pneumonia, sickle cell anemia, cystic fibrosis, and some forms of breast cancer. But I'm going to give you a spoiler alert. Most types of vasculitis are not familial single gene diseases. Instead, most types of vasculitis are complex genetic diseases. And these are diseases where the risk of disease is influenced by having mutations in many different genes. And each genetic change or each mutation contributes a very small amount to the disease risk. But it's not enough that you have these genetic changes to result in disease. You also need to have some type of exposure. So these exposures can be infections or drugs or diet or other exposures. And then there are other factors that can impact disease risk. So it's a combination of genes with environmental exposures with other factors that can lead to vasculitis. Now, kind of an example of something like that might be that I can use to illustrate this would be melanoma or skin cancer, where we know that patients who have a certain type of complexion, a lighter complexion, are at increased risk of skin cancer. So their complexion, their skin color, surprisingly, is actually influenced by over 100 genes. But these are not the only genes that can impact whether or not you develop skin cancer. Other genes such as DNA repair genes also influence this risk. But even if you're light complexioned, you may not develop skin cancer if you don't have any sun exposure. So environmental exposures impact whether or not you develop skin cancer, as well as other factors such as sunscreen use. So this is an example where lots of different genes in combination with environmental exposures and other factors influence your risk of disease. Now, many of the common diseases we think about, if not most of the common diseases we think about, are complex genetic diseases. So, for example, high blood pressure, strokes, cardiovascular disease, depression, and others are diseases where lots of different genes are involved, as well as environmental exposures and other factors. But even within these diseases, there's a small subset of individuals who have these diseases that are due to a single gene mutation. So even amongst those with high blood pressure, there is a small group of patients who have high blood pressure because of changes in a single gene. And this applies to vasculitis as well. I'll go into that a little bit more. Now, how do we identify these genetic changes, these genetic mutations that are associated with vasculitis? We conduct genetic studies. And if you look at a genetic study, most of them are done in this type of fashion. You get a lot of individuals who have your disease of interest, these affected individuals, and we call them cases. And you get a large group of people who don't have your disease of interest. We call them controls. We collect DNA from both groups, and we compare DNA from the cases to DNA from the controls to see if there are certain genetic variants or genetic mutations that are more common in cases compared to controls. And this has been done for all different types of vasculitis. I'm just going to go through one of these genetic studies that was published recently, published in June of this year in giant cell arteritis as an example of the types of results we can see and what we can learn about them. So in this study of giant cell arteritis, they examined DNA from about 3,500 patients with giant cell arteritis, and they compared it to the DNA from about 15,000 controls, so 15,000 people without giant cell arteritis. And they looked at over 6 million genetic mutations, DNA-based changes, to see which of those genetic mutations are more common in patients with giant cell arteritis. And this graph here is just a summary of what they found. So on this graph here, each dot represents one of the genetic mutations they looked at, and it's plotted based on what chromosome it's on. So 22 pairs of chromosome, 22 pairs of non-sex chromosomes in each human cell. So plotted based on the chromosome and where it's located on the chromosome. Where the dot is on this graph gives a sense of how strongly associated that mutation is with giant cell arteritis. So, genetic mutations here are very strongly associated with giant cell arteritis. And basically, we identify a threshold where we say all the dots above this line here are associated with giant cell arteritis. They're far more commonly seen in patients with giant cell arteritis compared to those who don't have giant cell arteritis. And we have what we call our top hits, the genes that have mutations that are above this line. And our top hits are in 5 areas. One is the HLA area, and then in 4 different genes, PLG, MFGE8, CCDC25, and VTN. And I'll go into these in a bit more detail. But in this study, what's also interesting is they identified 3 new genes that are important in the development of giant cell arteritis, the bottom 3 that I have mentioned here. So that's great. So we've now identified, 5 genes that are important in giant cell arteritis. But what does that really teach us? What do these results really mean? So the top hit is in what we call an HLA gene. This is on chromosome 6. This is actually of no surprise to us. So HLA stands for human leukocyte antigen. It's a family of genes that help the body identify cells from oneself, from cells from a foreign entity, whether it be bacteria or other individuals. HLA genes are actually the genes that are matched in organ transplants to help prevent organ rejection. So kind of, helps the body recognize self from non-self. And I say these associations with HLA genes are no surprise because the HLA genes have the strongest genetic associations with essentially with every single autoimmune disease, vasculitis included. So the HLA genes for giant cell arteritis are the top hit. But what we can see is that the odds ratio here is 2, which means that it increases, if you have this mutation, it increases your odds of developing giant cell arteritis about 2-fold, which seems like it's a lot. But I think you have to take this in context that the risk of giant cell arteritis in somebody who's over 50 years old is 1 in 500. So even if you have this HLA mutation here that increases your odds 2-fold, that means your risk increases from 1 in 500 to 2 in 500. So greater than the general population, but still pretty low. The next three genes on the list are plasminogen and lactadherin and vitronectin, very complicated names, just to say that they are involved in blood clotting and the development of blood vessels. And you may actually think, well, of course, this is a vasculitis. We would expect genes associated with blood and the development of blood vessels to be important in the development of vasculitis. But that's actually not the case. This is actually the first time we've seen genes involved in the development of blood vessels to be associated with vasculitis. Most of the time we see genes that are involved in immune responses associated with vasculitis. So this kind of gives us a sense that there are these other pathways and other cell activities out there which are important for the development of this disease. And then the last gene that was associated is this CCDC25, just to say that this gene is involved in the body's response to a bacterial infection. So this can give us a sense that maybe one of the environmental exposures that is important for the development of giant cell arteritis is the body's response to bacterial infection or bacterial infections. So I think this, I just wanted to give a sense of like, while the risk themselves imparted by each of these genes, so aside from the HLA, the risk, increase the risk, impact on disease risk is low. The genes themselves do actually inform us quite a bit about the disease and the processes that are important in the development of disease. Now, these are because the odds ratio, because the risk imparted by each of these genes is pretty low, it doesn't really make a whole lot of sense to try to test for each of these gene mutations independently. Like, it doesn't make sense to try to test whether or not somebody has this plasminogen mutation. But you may ask, well, Sharon, there are 5 genes here. What if we combine them all together? Like, what happens if we combine them all together? Is that a better estimate? Or does that better inform us whether or not somebody's going to develop vasculitis? And what that is what we call a genetic risk score. If we develop methods of combining the information we have from all of these different genes, if we add up all these different genes together, can that inform us whether or not somebody is going to develop vasculitis? And what we would love to see is what's shown here on the graph on the left. So let's say we develop this genetic risk score where we add up the number of different associated genetic mutations somebody has. It would be great if we saw that people who don't have giant cell arteritis have a much lower score than people who do have giant cell arteritis, who would have a much higher score. And we could use this genetic risk score to help diagnose patients. This is the ideal world on the left. What we actually see is what's on the right, where the histogram, the plot of genetic risk scores for people who don't have giant cell arteritis is in blue, and the plot of the risk score for patients with giant cell arteritis is on the right, is in orange. And what we can see is that the scores for patients with giant cell arteritis are shifted over to the right a little bit, but they're not so clearly separated such that, if you have a score that is this or below, you're unlikely to have giant cell arteritis. And if you have a score that's, a specific level or above, you are likely to have giant cell arteritis. The scores are not that well separated. So even if we tested patients for a whole bunch of different genetic mutations and combined that information together, it's still really not enough for us to be able to differentiate those who do and do not have giant cell arteritis. Now, this is a study in giant cell arteritis, and I picked this one because it's one of the ones that's been most recently published. It was published in June of this year. But this type of study has been conducted for all different types of vasculitis. So It's been done in Takayasu arteritis, which is another large vessel vasculitis. It's been done in the ANCA-associated vasculitis, so including GPA, MPA, and EGPA. It's been done in Behçet's disease, which is a vasculitis that affects all different vessel sizes. It's been done in cryoglobulonemic vasculitis. It's been done in IgA vasculitis, and a whole lot more. But the overall kind of ideas cut through all these different studies, which is the top hit in all of these studies tends to be on chromosome 6, which is the HLA. So these are the human leukocyte antigen genes that help differentiate between self and non-self. There are genes outside of the HLA region that are also associated with disease risk, but the risk imparted by each of these genes is low. So the takeaways, we've done genetic studies for a lot of different types of vasculitis. We've seen lots of mutations in a lot of different genes, and the overlap between different types of vasculitis is actually not very high. There appears to be genes that are important for one type of vasculitis, but it's not so important for another type of vasculitis. The risk conferred by each of these mutations is really quite small. And so because of that, it's really not useful to test individuals for the associated gene mutations to determine whether or not that individual has a particular disease or will develop a particular disease. So then you might say, okay, Sharon, we've spent all this effort doing all these genetic studies, and at the end of the day, we still don't have a genetic test that we can use to help diagnose patients with disease. Why should we be doing these genetic studies? And the answer is that these studies really give us insights into how diseases develop. What are the immune pathways that are important for diseases? What are the cellular processes that are important for developing disease? And so while, we may be disappointed that we don't have a genetic test at the end of the day, we still learn quite a bit about the different types of vasculitis when we do these studies. Now, remember when I said, for something like high blood pressure, there is a small subset of patients who have high blood pressure because of mutations in a single gene? The same can be applied to vasculitis, where for the vast majority of patients with vasculitis, their vasculitis is due to mutations in many different genes, each of them that confer a very small risk. However, there is a subset of patients with vasculitis that have their vasculitis due to a mutation in a single gene. And this is probably best illustrated by a process that we call DADA2, or deficiency of adenosine deaminase 2. This is a single gene type of vasculitis that was first published in 2014 by a group at the National Institutes of Health led by Dan Kastner. DADA2 is a process that can affect many different organ systems. The two most dramatically affected organ systems are your brain, the central nervous system, as well as your skin. And what can happen for patients in the brain is that they can have these strokes, these pretty dramatic strokes that can damage large parts of the brain. These patients can also have very dramatic skin rashes, shown here on the bottom right, and other organ systems that can be involved and can include their kidneys, where they have high blood pressure or protein in their urine, can be in their GI tracts, where they can have tissue damage of their bowel, or can actually even affect their testes and have testicular pain. And I bring all of this up because these are actually the same symptoms that we can see in another type of vasculitis called polyarteritis nodosa. And so many patients with DADA2 were initially diagnosed as having polyarteritis nodosa. So DADA2 is a subset of patients with polyarteritis nodosa where their disease process is caused by mutations in a single gene. That gene is adenosine deaminase 2. If you have mutations in adenosine deaminase 2, you have decreased activity of the adenosine deaminase protein. Some patients have no activity of this protein, and some patients have a little activity of this protein. And if you can't, if depending on how much activity of this protein you have, it impacts whether or not your body can break down adenosine. And if you can't break down adenosine, that results in inflammation. Now, DADA2 is a single gene disease that is inherited in what we call an autosomal recessive pattern. So in that situation, each parent has one copy of an ineffective gene, but they themselves are doing okay because they have another copy which is functioning normally. What they do is they pass on their defective copy of the gene onto their child, and then their child has decreased activity of the adenosine deaminase protein. Now, as a single gene disease, most patients are diagnosed as children, but some can be diagnosed as adults. 25% of patients are diagnosed by 1 year of age, and about 3/4 of the patients are diagnosed by 10 years of age. But there are patients who are diagnosed in their 20s or 30s or 40s or older. And the way we diagnose them is we do testing for either the enzyme level for the ADA protein, or we sequence the ADA2 gene. Now, this is a genetic single gene disease. It's actually pretty rare. So there have been 600 individuals with DADA2 identified. I think you can compare that to, for example, the more than 10, if not more than 15,000 patients in this country alone who have something like granulomatosis with polyangiitis or microscopic polyangiitis. So there are there are single gene causes of vasculitis, but it's really a small subset of the total group of patients with vasculitis. I think another interesting, so just some takeaways from this. So there are vasculitic syndromes caused by single genes, but these vasculitic syndromes are pretty rare and more likely to present at younger ages. We think about DADA2 as kind of like a single gene subset of polyarteritis nodosa. And what does that mean for my practice? So in my practice, I test patients suspected to have polyarteritis nodosa for DADA2 mutations. And the reason is that it impacts treatment decisions. So if you're a patient with polyarteritis nodosa, I'm more likely to treat you with medications such as cyclophosphamide or methotrexate or azathioprine. However, if I find that actually you have the DADA2 type of polyarteritis nodosa, then I am more likely to treat you with another family of medications, called TNF inhibitors, because these classic medications, the cyclophosphamide, the methotrexate, the azathioprine, they're not likely to work for you. All right. Let's go here. So I think there's been a lot of interest in this next group of vasculitis. So this is also a type of vasculitis that's due to a single gene mutation, but it's actually due to an acquired mutation. So what does that mean? Sorry, I've lost my gift of talk today. So the vasculitic process that was first identified to be due to an acquired mutation is called VEXAS. It's this VEXAS syndrome, and it was first published not that long ago in 2020 and 4 years ago. So VEXAS is an acronym, and it's an acronym for what we see in this disease process. The V stands for vacuole, so patients develop vacuoles or holes in the cells in their bone marrow. It's due to mutations in this ubiquitin activating enzyme called UBA1, which is located on the X chromosome. If you have mutations in UBA1, you have autoinflammation. Your body produces inflammation inappropriately, independent of known triggers. And this process occurs by somatic mutations. So I'll get into that in a minute. So patients with VEXAS have fevers and fatigue and low blood cell counts. They can have inflammation of the ear and inflammation of the eye. They can have particular different types of skin lesions, including medium vessel vasculitis of the skin. Patients with polyarteritis nodosa, which is a medium vessel vasculitis, relapsing polychondritis, which is a process that involves the ear and the big blood vessels in the body, and giant cell arteritis have subsequently been recognized to have VEXAS. So this is, another area where small pockets of patients that have been diagnosed with a particular vasculitic process have subsequently been recognized to have VEXAS. Now, I say VEXAS is caused by a somatic mutation. What does that really mean? So for the genetic mutations that we saw that were associated with giant cell arteritis, or the genetic mutations that are important in DADA2, these are due to what we call germline mutations. These are genetic mutations that are present in the very first cell of the embryo. So they are present when the egg is fertilized by the sperm. Because it's present in the very first cell of the embryo, this mutation is in all of the cells of our body, and this mutation can be passed on to our children. VEXAS, on the other hand, is caused by a somatic mutation. So this is a mutation that is not present in the first cell of an embryo. This is a mutation that we acquire at some point in our life, and it persists. And because we acquire it later in life, it's only present in some of the cells of our body, and the mutation is generally not passed on to our children. So as we go through life, our DNA acquires genetic mutations, and for the most part, those genetic mutations don't have a significant impact. And that can be shown in the blue cell here, which, has a genetic mutation, but it really doesn't do anything. And that cell kind of percolates along until it dies. On the other hand, we can have a cell like this green cell here, where the genetic mutation occurs at some point in our life, and this genetic mutation persists. The cells divide, and the subsequent cells all carry that genetic mutation. Now, as I said, DADA2 and the genetic mutations that I talked about for giant cell arteritis, those are germline mutations. They're in every single cell of our disease. But these acquired mutations, depending on when they occur in life, they can affect large parts of our body or very small parts of our body. In VEXAS, what has happened for these patients is that these patients have acquired mutations in the UBA1 gene in immune cells. So this mutation has occurred specifically in immune cells, such that these immune cells still persist, and it leads to uncontrolled or inappropriate inflammation. The idea of somatic mutations causing a disease is actually not new. It's actually the same mechanism that develops in cancer. So what happens in cancer is that at some point in our lives, there is a mutation. We acquire a mutation in genes that control cell division. And because of this mutation, we have uncontrolled cell growth. And that's essentially what cancer is. Cancer is a group of cells that have acquired mutations in genes that control cell growth, such that they no longer respect the boundaries for cell growth and they keep on growing in places where they shouldn't be growing. So both VEXAS and cancer are due to somatic acquired mutations. I'm just going to put a pause on people here. I don't want people, to go on social media and say vasculitis is a cancer. Vasculitis is not a cancer. It's just that these two types of diseases can use the same mechanism to develop. So what do we take away from this? So we all acquire genetic mutations over time, but don't panic. It's okay. Most of the time, these mutations occur in places that really don't have an impact. Rarely, these mutations can lead to autoimmune diseases, including vasculitis and including processes like VEXAS. So what does that mean for me in my practice? So I will obtain this UBA1 gene sequencing in certain situations. So, for example, I said that there are patients with giant cell arteritis who have been subsequently found to have VEXAS. This doesn't mean I'm going to test every single patient with giant cell arteritis to see if they have VEXAS. But if their, type of giant cell arteritis is a little unusual, so for example, they're having really low blood counts or they're having unusual skin rashes, kind of the manifestations that we see in VEXAS syndrome, then I think it's worthwhile to test patients for these UBA1 mutations. And the reason I do it is similar to the reason I do it for DADA2 and polyarteritis nodosa. Patients who have VEXAS likely should undergo different treatments, and they could be considered for bone marrow transplants, which can be curative. All right. So, I can hear people thinking, Sharon, that's fine and dandy. The science is really interesting. But really, what I'm interested in knowing is whether or not vasculitis is genetic. And what that question means to them is, does vasculitis run in families, and will my children get it? Do I need to be worried about whether or not my children will develop vasculitis? And the answer, unfortunately, or I would say the question, unfortunately, is not very well studied, especially here in the US. I'll be honest, we don't have a national healthcare database where information about every individual is entered. And because of that, we have a hard way of tracking who has what disease and how often it's occurring. There are case reports of parent-child pairs or siblings with vasculitis, but these case reports are actually pretty rare. This question is actually better studied in countries who do have the national kind of healthcare database, and I'll give you some examples of what we've identified. So some of the best information that we have is actually focused on Behçet's disease. So this is a vasculitis that affects multiple different types of blood vessels in the body. It's associated with eye inflammation as well as oral and genital ulcers. This is a disease that's far more common along the old Silk Road. So in Turkey, where this disease is far more common, if you are an individual with Behçet's, your sibling has an 11-fold to 50-fold higher risk of Behçet's compared to the general population. That sounds pretty high, 10-fold, 50-fold. But the overall prevalence of Behçet's in the population is about 1 in 1,000. So that means the risk for your sibling, the risk for your brother and sister is about 1 to 5%. So it is much higher than the risk of the general population, but it's still pretty low. In Korea, your risk of developing Behçet's is about 13 times higher than the general population if one of your parents is affected. It's about 15 times higher if one of your siblings is affected, and it's about 165 times higher if your twin is affected. I think that's understandable. Your twin shares your genetic DNA, and so, your risk is going to be higher if your twin is affected. But the overall prevalence of VEXAS in Korea is really quite low. It's 35 per 100,000. And so even if if you yourself have VEXAS, the risk to your child is about 0.5%. So your child has, the likelihood that your child is going to develop VEXAS is 0.5%. So higher than the general population, but still pretty low. So for Kawasaki's disease, which is a medium vessel vasculitis affecting the heart, primarily occurring in children, your risk of developing Kawasaki's is about 10 times higher than the general population. If your brother or sister has it, it's about 2 times higher than the general population if your mom or dad had it. So the prevalence is about 1 in 100 in Japan. So if you have it, there's a 10% chance that your brother or sister would get it, and there's a 2% chance that your child would get it. And then for GPA, granulomatosis with polyangiitis, a small vessel vasculitis affecting primarily the lungs and the kidneys. The study that we have comes from Sweden, and what they showed is that your risk of developing GPA is about 1.5 times higher than the general population if you have a first-degree relative who has GPA. So if your mom, your dad, your brother or sister has GPA, your risk is 1.5 times higher. So that means your risk increases from about 60 per million individuals to 90 per million individuals. So yes, your risk is higher, but the risk remains quite low. This study was actually interesting in that they looked at diseases besides vasculitis. And what they saw is that there's an increased risk of autoimmune disease, of any autoimmune disease in the first-degree relatives of patients with GPA. So your risk of any autoimmune disease is about 30% higher than the general population. And your family members' risk of specific autoimmune diseases such as multiple sclerosis or Sjogren's syndrome or rheumatoid arthritis ranges from about 1.5 to 2 times higher than the general population. So, when I'm asked, is vasculitis genetic? What I generally say is that the chance that your family member will develop vasculitis is higher than the general population, but it's still pretty low. And so I think, you and your family members should go out and do the things you want to do in life and not worry about whether or not your brother, your sister, your mother, your father, your child will develop vasculitis, because the likelihood that they will develop vasculitis is very low. Your family member may have an increased risk of developing another autoimmune disease compared to the general population, but that risk still remains low as well. So I don't think anybody should be panicking about what's going to happen to their family members because they themselves, with regards to vasculitis, because they themselves have been diagnosed. So to answer the question, yes, vasculitis is genetic, but the chances that your family members will develop vasculitis is pretty low. Are there genetic tests that should be sent? And I've touched upon this in the presentation. I think there are certain situations where, I would send off sequencing or testing for specific genes, such as ADA2 for DADA2 or UBA1 for this VEXAS process. And that is if the clinical picture fits one of these diseases. But in the absence of this type of situation, there isn't really genetic testing we would send off to diagnose a particular type of vasculitis, such as giant cell arteritis or granulomatosis with polyangiitis, or etc. However, I do consider sending genetic testing to prevent medication toxicity, and this is particularly in regards to azathioprine. Azathioprine is an oral immunosuppressant that we used to use pretty commonly for the ANCA-associated vasculitides. We use it pretty commonly in to treat other autoimmune diseases such as systemic lupus, etc. We know from prior studies that there are two genes. One of them is called TPMT, and the other one is called NUDT15. These genes code for proteins which help break down azathioprine. And in a small proportion of individuals have mutations in these genes such that the enzymes don't work very well and the azathioprine is not broken down. And if the azathioprine is not broken down, then it accumulates in the body and can lead to toxicity such as very low white blood cell counts. So if I have a patient that I'm considering starting azathioprine in, I will send off testing for TPMT and NUDT15 to see if they have these mutations, because that influences whether or not I start them on azathioprine, and if I do, the dose that I use for patients. Okay, so in summary, vasculitis can develop due to the effect of multiple genetic mutations, so what we call genetically complex diseases, or mutations in a single gene that can be germline, so present at conception, or somatic, developed over time. When it's a genetically complex disease, as most of our vasculitides are, the risk imparted by each of these genetic mutations is really quite low. Because of that, most of the time we don't do genetic testing to diagnose vasculitis, and the risk of vasculitis in family members of affected patients is really low. There are a few genetic tests that we utilize clinically. So as I said, the genetic testing that I do for azathioprine and the genetic sequencing we can do for DADA2 and VEXAS. I think that's all I have planned to cover today. I really want to thank you for your attention. This is always a fun thing to think about. I want to thank the VF Foundation for giving me the opportunity to talk today, and for the thousands and thousands and thousands and thousands of patients that have participated in these genetic studies so that we can learn more about your disease. So thank you very much.
So thank you, Dr. Chung. You can stop sharing so that we can
Will
just
do. Yep.
see you. Great. We got quite a few questions while you were talking. Some of them you answered in your presentation, but there are a few very interesting questions. So in the interest of time, I'm going to get started right away on that. I did want to remind everybody, if you put a question in the question and answer box, please remember that it needs to be specific to genetics today. Although your questions are all important about your disease, we're talking about genetics today. The first question is, where is most of this genetic research happening?
So this genetic research is happening all over the place. So a lot of these genetic studies came from the United States and through efforts of something called the Vasculitis Clinical Research Consortium. This is a group of investigators across the U.S., actually across North America, that have banded together to conduct a number of different studies, not just genetic studies. There are investigators in Europe who are very interested in this. There are investigators in Asia who are very interested in this. As you can see, we need large groups of patients to do this, thousands and thousands and thousands of patients. So these studies only happen in collaboration between investigators all across the world.
Okay, thanks. And this, interestingly enough, somebody did post in here, my husband and his brother have GPA. Should they be tested?
So I would say yes. What I would recommend is actually to see if you can be seen at one of the sites that participate in the Vasculitis Clinical Research Consortium to see if we can get your samples. That would be great.
Yes. Someone says if gene mutations contribute to certain types of vasculitis, does that mean that gene therapy could be used in the future to help treat or prevent forms of vasculitis in at-risk patients?
I think that's a very interesting question. I think I'm going to say probably not, because if you think about it, you know, most types of vasculitis are due to mutations in a lot of different genes. So, 5 genes, 10 genes, 20 genes, if not more. So, fixing the mutation in one gene is unlikely to have a significant effect. And I think it's very complicated to fix multiple genes. But not only that, as I said earlier in my presentation, you have genes and you have environmental risk factors. So just fixing genes alone is probably not enough to completely get rid of your risk of vasculitis.
That makes sense. Someone says, and I'm not sure I'm going to pronounce this correctly, but can DADA2 be related to the Factor V Lieden
Lieden, Factor V Lieden, yeah. So DADA2 and Factor V Lieden are completely different genetic mutations. They're in different genes. So they're, I would say, almost with certainty not related.
Okay. And interesting, this person says, my mom is diagnosed with UV and I'm diagnosed with HUVS. My doctor tested me for mutation in DNASE1L3. Test was negative. Is there any other test I can take if this is genetic, or do I just have bad luck?
I think so. UV is urticarial vasculitis. HUVS is hypocomplementemic urticarial vasculitis syndrome. The DNase 1L3 gene is one that's been associated with this, but there are also other genes that have been associated with these processes. But as I said, the genetic mutations aren't strong enough. The associations aren't strong enough to have those tests be used in the clinic quite yet. So I suspect these diseases are genetic in that genes influence their risk, but we don't have tests for them yet.
Okay, and I think you answered some of this, but maybe they just need a clarification. If you develop an autoimmune disease but do not have vasculitis, but have a parent with vasculitis, does that autoimmune disease put you at a higher risk of developing vasculitis?
No. So, I mean, the example would be like, let's say a patient has granulomatosis with polyangiitis, GPA, a small vessel vasculitis. They may have a family member who has, for example, rheumatoid arthritis. That person with rheumatoid arthritis doesn't have a higher risk of developing GPA, developing vasculitis, etc. Like, I would not expect that person with rheumatoid arthritis to develop a vasculitis on top of that.
Okay. And I know we've all heard, and this patient is asking, I've heard autoimmune diseases run in families, but individuals might get different ones. Has that been studied?
Yeah, so that's what was shown in the study in Sweden for patients with granulomatosis with polyangiitis. Their family members have a higher risk of developing other autoimmune diseases like rheumatoid arthritis, multiple sclerosis, etc. And we actually know that for not just vasculitis, but other autoimmune diseases such as lupus or rheumatoid arthritis, that patients who have lupus or rheumatoid arthritis, their family members also have a higher risk of other autoimmune diseases.
Okay, and this might be a huge question. What makes genes mutate?
Yeah, so I think a lot of different things make genes mutate. So your DNA codes for, your DNA contains the genes. I think of your DNA as a set of encyclopedias or a set of books that have instructions and all of them. And each time your cell divides it has to recopy this set of instructions. And if you just think about, like, if you write the same sentence over and over and over again, sooner or later, you'll make a mistake. And it's not intentional. It's just, something that happens, that there is a mistake introduced into a gene. That's one of the ways that genes can mutate, is that just as cells are dividing and the DNA is being copied over and over and over again, there's a slip up in the DNA machinery and the wrong base is inserted and a genetic mutation occurs. So that's one way it occurs. Another way that it occurs is that for example, there are chemicals that can mutate DNA. There are environmental exposures such as UV light that can mutate DNA. There are a lot of different mechanisms that can mutate DNA.
Okay, and I think one final question. This person is asking, is it just a coincidence that I have PMR GCA and my brother's son has Behçet's disease?
I have to look at the comparison between genetic associations between the two. I'm not sure coincidences. I think this falls into the idea that autoimmune diseases run in families and different family members have different types of autoimmune disease. So I'm not sure if coincidence is the right word. I think it's just another sign that autoimmune diseases run in family.
Right. Well, we definitely would like to thank you so much, Dr. Chung, for sharing your time with us today and answering all these questions. And your presentation was fascinating, and I appreciate it so much. And we'd like to thank the Vasculitis Foundation for sponsoring and making these webinars possible, as well as our sponsors, Amgen, AstraZeneca, and Novartis. And thank you, of course, to Dr. Chung and to all of you that asked these great questions. It was very informative today.